And is my PPT visible to you all? Okay, so welcome to this Developmental Biology Unit 5, and we are discussing all the previous year CSIR NET question. Here is your first question from Subunit 5B, which is regarding fate maps. Okay, yes, good evening, nature. Given below are fate map of two organisms and the pattern by which embryos undergo cleavage. Which of the following are right combinations? So this is the question. As you can see, here we have the animal and the vegetal pole, okay, of different organisms which are given. Now all of these are different organisms, and after the embryonic cleavage division, cleavage, as you would know, it's a cell division. After that, they would give rise to certain kind of structures, okay. And if you would know the different types of cleavage divisions that we have, discoidal, rotational, superficial, radial, you have to connect with which one is properly combined, okay. So for that you have to know this fate map. Actually, these are the fate maps which are showing different parts of the organism, how they look, okay. And how, which structure is going to form which part of the in the later stages, which part of the organism. This is what is the diagram of a fate map. Now if you would see, this is what a fate map is. This chart or graphical representation detailing the fate of each part of an early embryo in the adult animal is referred to as a fate map, okay. So this is one of the zebra fish, in which you can see the top portion is ectoderm, and the mid portion would be forming endoderm, and then the bottom portion it would form the endoderm. Okay. Yes, Anusha Nelima, very good evening. And then frog, you can see there is a typical structure like this over here, and so this is what is going to form the notochord. Okay, so notochord structure is like this. Whereas in mouse, you would see the notochord structure is a small area in the early embryonic stage, and in the chick, it is like a semicircle. So you can remember by different things. Also, the neural ectoderm is covering a small area in the chick, whereas in mouse, it is slightly larger area. In frog, it is almost half of the ectoderm, and in zebrafish, it is not visible. So by this only, you can figure out which is what. So as you can see, this is of a amphibian. Just now we saw this is frog, representing frog, toad, salamander. So this is amphibian. Okay. And then I told you that notochord part in mouse is very small. So as you can see, this is for the mouse part, because in chick anyways you would see it as a semicircle, okay, half semicircle. It is done in the early fate map. So this is also chick embryo. Again, this is amphibian only, this is chick. Now, regarding the type of blastula that we have, we know that for chick, we have the telolacethal egg, which means the entire egg bottom portion, or the vegetal portion, what we call, will be surrounded by yolk, and the cleavage division will happen only at a non-yolky region at the top of the egg, and that will be called as the that portion would be yolk free, and if you look from the top, it would look like a disc. That is why it will be discoidal cleavage. Okay, so chick has discoidal cleavage, type of cleavage. It is meroblastic, which means the entire egg will not be divided, only a portion of the egg is going to be divided. That's why it would be meroblastic. Okay, whereas so which are the right combinations you have to mark over here. So you have the chick going for discoidal. Anyways, chick is not discoidal. So we would be sorry, the mouse will not be discoidal for the chick. Okay, so this is for the mouse. So anyways, chick is discoidal, chick is not shown over here. So means fate map is not shown, but the cleavage division, as you can see, it is happening only at a topmost portion over here. So that is why this would be of chick. But since I said this is for amphibian fate map, that is why this is not a correct match. Okay, then mouse, we know it is mammals. So mouse we have rotational holoblastic cleavage, in which the first cell division will cleave the egg into equal halves, but the next cell division there will be only one, and one cleavage plane will be in different plane, and the other cleavage division will be in another plane. So the two cleavage divisions will be in different plane, and that type of cleavage division is called as rotational. So both of this will be for mouse, which is a type of mammal, and that is why this is correctly matched. Okay, so B is correct match. Again, amphibian they have given superficial, so this is again not correct, because superficial cleavage is a characteristic of insects. Okay, that is the example Drosophila, because there we have the centrolecithal egg, which means the yolk covers the entire central area, and the peripheral area around the yolk, it will whatever will be free there, only the cleavage division will be taking place. So this is what is the superficial cleavage happening in insects, but since this is amphibian fate map, this is also not correctly matched. And anyways, mouse I have told you it is rotational. So radial is not the answer. Radial is in fact for amphibians. Okay, so these are also not correctly matched. Amphibians have mesolethal egg, which means they would have a little bit amount of yolk in the bottom vegetal portion. Still the egg will completely divide, and this is what would be called as a holoblastic cleavage, but it would be radial, means there would be either top to bottom cleavage division, that is from animal to vegetal pole, or perpendicular to the animal to vegetal pole. Okay, so that's why B is the correct answer here. Okay, this is regarding sea urchin. You can answer this regarding sea urchin. As you would know, in sea urchin, you have a specific type of cells which are present at the vegetal pole, and those vegetal pole cells, those are called as micromeres. Okay, and that too, these are the large micromeres. So, what are large micromeres? These are they would be large micromeres. They are supposed to give rise to the axis formation. Okay, and in sea urchin, they would be deciding the fate of the embryo. So here you would have to know that in large micromeres, a particular type of marker is there. That means a particular type of factor is expressed over there. Okay. So kindly solve this question. For example, we would be having this HESC. Okay, HESC is a type of factor which would inhibit the transcription factors present in the vegetal section. Okay, in the vegetal section, there would be some transcription factors. So that would be inhibiting the transcription factors to make the skeletal micromeres. Actually, large micromeres are supposed to make the skeletal rods. Okay, so that is why some kind of transcription factor should be expressed in the skeletal rods. So this diagram what they have shown, it is actually from the vegetal area. From the bottom they have shown. Okay, that means these are only the large micromeres. Okay, so they have not written over here what is what, but these are a category of cells. These are large micromeres, and the cells which are present around it, they would be the vegetal cells. So the rest of the cells which are present, those are the vegetal cells, and there are some factors which are expressed in A and some factors which are expressed in B. So as I told you, these large micromeres, they are supposed to form skeletal rods. So certain transcription factors should be activated if these structures have to form the skeletal rods. Okay, now there is a inhibitor which is called as HESC. The HESC inhibits the transcription factors. So what transcription factors is there? One is delta, then some more are there, transcription factors which would be making the specification of this particular area. So only delta is set. So you can see one of the genes controlled by HAS is delta, whose expression is used as a marker for micromere lineage. Okay, but the other vegetal cells, there we don't have this HESC. Okay, why? Because so what happens is that if HESC is present, let me tell you, HESC is actually an inhibitor of delta. Okay, HESC will inhibit the delta not to form. Okay, so delta is required, transcription factor it is required to form micromere. Okay, so what stops HESC is another factor which is called as PMAR1. Okay, so PMAR1 inhibits HESC, HESC inhibits delta. Delta is the transcription factor for micromer formation, micromer specification. So, in these micromeres, we want PMAR to be active. Okay, we don't want HESC. So, PMAR being present, it will inhibit HESC and HESC will inhibit the delta in these A category. Okay, so basically, you will not find HESC over here. If HESC is present, then they will not allow for micromer specification. Okay, so you can look here, they have used some hybridization technique, in situ hybridization, whole mount in situ hybridization using delta probe. So what is the probe over here? You would know hybridization concept. So there would be the gene, right, and the probe would be for binding to a particular section of the gene. So that's what happens. Now the probe is for delta, isn't it? No, yeah. So they have used the probe for delta, right? So let's say you have the delta gene, if you have, okay. So the delta probe, if it is binding, okay, that means the delta gene has been synthesized. Delta gene has been synthesized in the cell. So we want to know whether in micromer the delta would be there or not. So yes, in the micromer delta will be there, but in the nearby cell, in the vegetal cells, they are not micromeres. So there the delta gene will not be expressed. Okay, so if you do hybridization in these micromeres, then the probe will go and bind to the gene. That is how hybridization will happen, and you can visualize that hybridization. But around the surrounding areas, the vegetal cells, delta will not be there, because here we don't have PMAR1. PMAR1 is present only in the micromere, so that they can inhibit HESC, and HESC can not inhibit the delta factor, and delta factor can be expressed for micromer specification. But in the surrounding areas, that is not the case. So PMAR won't be present, so HESC will be present, and that is why HESC will inhibit the delta formation. So what it is saying, the whole in situ hybridization, if it is done with HESC probe. So here, this is the result for probe used with delta probe. Okay, so you can see purple color means the probe has gone and bonded, right? So in the central micromeres, you will see delta probe is showing, which means that in this micromeres delta is present. That means what? These are the micromeres. In short, they are giving you the hint that these are micromeres, this A group of section. Now they have given different experiments and where the coloration will be visible. That means where the probe will go and bind. So this is an extremely hard question from CSIR, but you can expect one or two questions like this. You can expect. So that's why we are discussing it here. Okay, so only if you have a clear understanding of the concepts, which you can get from any coaching class, like Paratechnica conducts the classes. Okay, so W, because we discuss everything in detail. Now here we have this in situ hybridization with HESC probe. So if we have the HESC probe, means what? Whichever cell is expressing the HESC probe and is not inhibited, so there you will see this purple coloration. Okay, so we know that in the micromeres, HESC probe is inhibited, isn't it? HESC is inhibited. So here in A, you will not see coloration. Okay, but in the nearby areas, PMAR is not there. So there HESC can be present. So you will see this in B. Okay, the A you would be seeing in 2. A would be 2 actually. Okay, this 2 is for B. So A would be 2. Okay, so that means it can be either 1 or it can be option 2. Now let's take one more for what about B? The in situ hybridization is done with delta probe. Okay, same like this result, same experiment, but micro injection of PMAR1 mRNA into fertilized eggs. Now see, PMAR1 naturally it is supposed to be present only in A. That means only in the micromere. It is not supposed to be present in the nearby vegetal areas. Okay, but what you have done is in the fertilized egg you have micro injected PMAR1, because of which now PMAR1 would be present everywhere, isn't it? It would be present everywhere. That is why HESC will not be present anywhere, because PMAR1, if it is present in all the cells, in A also, in micromeres also, in vegetal cells also, A also, B also, it is present, means this PMAR1 will be inhibiting HESC. So HESC is not present anywhere in the embryo, which means that delta will be transcribed in all the cells of the embryo, because HESC was inhibiting the transcription factors for micromeria specification, that is delta. So if PMAR1 is not there, if PMAR1 is there, HESC will not be there in any of the cells. So delta will be synthesized in all the cells. So if you perform hybridization with delta probe, you will find coloration in both A and B. So A, B will be option 3. Okay, so here also you can see B is 3. Now for C, you have to select either it is 3 or it is 4. So again hybridization with delta probe, but now you have used an antisense of HESC into fertilized eggs. Okay, so antisense of HESC means now it is the same experiment as this only, because anyways if you put antisense of HESC, even if in some cells, that is the vegetal cells, even if HESC is present, it will not be able to be expressed. Okay, so that is why the probe can if HASC is not present, then delta can be transcribed in all the cells, because you have injected antisense of HASC in the fertilized egg, means it would appear in all the embryonic cells, right? Because embryonic cells come from cleavage division of the fertilized egg only. So that is why you will not find HASC in any of the cell. That is why delta will be able to be transcribed in all the cells. So C also will be 3. Okay, so that is why option 1 is the right answer. So here is the explanation, and this is what is called as a double negative circuit, because PMAR1 inhibits HESC and HESC inhibits the transcription factor. So as I was telling, delta, ETS1, TBR1, these are all transcription factors which are required for the synthesis of micrometer specification, which would ultimately lead to the formation of the skeletal rods, and this would take place in the micromeres, whereas in the rest of the embryo, on any of these factors will not be synthesized. Okay, so there is one more question, you can try answering this also. So I have already explained the first two questions. Now I want correct answer from all of you. This is from CSIR September 2022 regarding C. elegans axis specification. It's regarding the signals and specifications actually which cells give to each other. Anyone has studied this C. elegans specification? Tell me. Okay, nature has said two. Let's see if anyone else wants to answer. Read the options and you can use elimination technique. Anyone else wants to answer this? Okay, let me take up this question then. The signals from the P1 blastomere. See, we all know in C. elegans, we have the anterior to posterior, the presence of AB cell. This first cleavage division is asymmetric, which produces a big AB cell and a small P1 cell. Okay, this is what we get from the P0 zygote. When the fertilized egg forms P0, then this is what it divides into. Okay, then they further will divide. The C. elegans specification division is also rotational. So AB cell divides into two daughter cells by a horizontal division, that is equatorial cleavage division, and P1 cell divides meridianally. So P1 also produces two daughter cells, but the size of the daughter cells, because of that, they would shift their places. So that is why they would form the cells which would be uh, one daughter cell of P1, which is P2, will be present here. Another daughter cell of P2, it would be EMS, which is present here. And then one of the daughter cell of AB, it would shift over here on top of the EMS, and with signal by P2 gives signal to this daughter cell of AB to form ABP, and the cell which is present towards the anterior, it would still form the ABA, that is the anterior most AB daughter cell. Okay, so you must learn all the cells what they are giving rise to from your theory classes. But as of yet, for this question, you should know that the signals from the P1 blastomere instructs the EMS cell for its further development. So this is not correct. You have to mark the correct questions, because E2 actually in P2 instructs the EMS daughter cell, so that the daughter cell which EMS will form, okay, EMS will also divide. So one of the daughter cell, actually I should write MS and E. So the daughter cell which is closer to P2, that differentiates into E cell. So since it is written P1 instructs EMS cell, that is not correct, because only when P2 is made, then only the it can signal the EMS. So it should be P2. In the absence of this instructive signal, EMS cell will divide into two MS cell. So what happens? I told you P2 gives signal to this one of the daughter cell of EMS to form endoderm. So that means if the P2 signal is there, then this daughter cell forms endoderm and the other daughter cell will form mesoderm. But what if P2 signal is not there? Then the daughter cell which is supposed to form endoderm, that will also form mesoderm. So that is correct. In the absence of the instructive signal, EMS cell will divide into two MS cell. So B is correct. Okay. In the absence of the POP1 signal. So what happens is when P2 gives signal to the E cell, in the E cell, the cell which is supposed to form E, a molecule called as POP1 is down regulated. Okay, so that is why if POP1 is down regulated, then only the cell can take endodermal fate. But if POP1 is present, like in mesodermal cells, in those cells, endoderm fate cannot be taken and the cell goes for mesoderm specification. Now, what if POP1 is not there? Then, even in MS cell, if POP1 is not there, then MS cell can take up the endoderm fate, because POP1 inhibits the endoderm fate. So P2 gives signal to inhibit the to downregulate POP1 in future endoderm cells, because of which it cannot form the MS. Okay, because of which POP1 inhibits the MS pathway, and that's why endoderm forms. So if you don't have POP1, the prospective MS cell cannot go to MS cell and it would take up the E fate. So that is why C is also correct. So B and C, option 2 is the right. MOM2 protein, it is the signal released from P2 neighboring the EMS cell, that is P2 present near the EMS cell, instructs the dividing EMS cell to take up the MS fate. So this is again not correct. Just now I told you it would instruct the dividing EMS cell to take up the endoderm fate and not the MS fate. Very good, nature. Your answer is correct. Okay, so this is what I was telling you. The signal name is MOM2 and the receptor name is MOM5. So P2 releases signal MOM2, and the EMS daughter cell has MOM5 receptor, because of which they will downregulate the POP1 and lead the daughter cell to the formation of endoderm, whichever is closer to P2, because this is a paracrine signaling. It cannot signal the cell which is away from it. Okay. Then one more question. This is regarding amphibian development. Anyone wants to try to answer this question? So as you would know that in amphibians we have the dorsal ventral axis formation done by the organizer, Spearman's organizer. This question is regarding activin and goosecoid and XBRA. So XBRA is present in the cells which are supposed to give rise to the organizer. Okay, and actually XBRA gives the mesodermal fate. Okay, and goosecoid will be giving rise to the organizer, the cells expressing goosecoid. So based on that, they have given an experimental question. Try to answer, else I will explain. Okay, so have a look over here how this question will be done. So we have the, I'll show you the diagram first, right? So this is what the question is asking. We have the activin bead, okay? So when you take 1 nanomolar activin containing bead in the center of the cells, so the wherever the bead is present, okay, and we can see that around the bead we have the XBRA expression in the cells near to the bead when we have taken 1 nanomolar activin. Now 1 nanomolar is small quantity, but when we increase the activin, when we take 4 nanomolar activin containing beads, now in this nearby area, okay, we don't see the expression of XBRA, okay, when just around the bead. Because why? Because in this region there would be high concentration of activin. Since it is 4 nanomolar, no, the concentration would be high, and in the nearby areas there would be high diffusion, okay. But in these areas, so in these areas you will see less activin concentration because it would diffuse out, no. So that's why this diffusing out will be in a, so what happens is in this area only, the when it is diffusing out in this area it will be more, but as it diffuses out concentration will be less. So that is why in this area the concentration would be less, okay. And anyways when it was 1 nanomolar then also concentration would be less because 1 nanomolar is extremely small concentration, okay. So when nanomolar is low concentration, less activin was diffused in the nearby cells. But in 4 nanomolar concentration, the distant cells would get less activin when it diffuses from source bead. So thus only the distant cells could activate XBRA and not the nearby cells. So only the distant cells here you would see the XBRA activation and not the nearby cells. Okay. So why this happens is because the activin, so what happens is not the nearby cells which received high activin concentration, because activin activates goosecoid and goosecoid inhibits XBRA. Okay, so wherever activin is there, that is in the nearby yellow color region, you will see that goosecoid is present because activin is high over there. So only high concentration of activin can activate goosecoid. Okay, so only where activin is present, there you have the inhibition of XBRA. So in this yellow portion, XBRA is not expressed. Okay, whereas the distant regions where you have low concentration of activin, so there you don't have goosecoid, because of which there you would see the XBRA being synthesized. Okay, so hence it can be concluded that in xenopus embryos, activin induces a range of mesodermal genes in a concentration dependent manner. The genes XBRA and goosecoid are activated at low and high activin concentrations, and 2 hours post bead removal. Even if you remove the bead, but already you can see the effects are still there, right? So what they have asked over here is high concentration of activin activates goosecoid. That is true, whereas low concentration activate XBRA. That is why A is correct. So A is correct means B, what it is saying, low concentration of activin helps specify dorsal ventral structure of frog embryos. Why? Because dorsal ventral axis means, so dorsal ventral axis, we know it is specified by goosecoid, isn't it? So if goosecoid is activated, okay, then dorsal ventral structures will be formed. But for high activation of goosecoid, we need high activation of activin. But it is saying low concentration of activin helps specify dorsal ventral. So B is not correct. Again, C is correct because concentration of activin that do not lead to expression of the two genes specifies the cells to become blood vessels and heart. These would be formed from the ventral mesoderm. Okay, so obviously if concentration of activin is less, then it will not lead to the expression of goosecoid, and that would lead to the formation of blood vessels and the heart. So when animal caps are treated, see, animal caps are treated with this much of activin, they differentiate into ventral mesoderm, which includes mesenchyme and hematopoietic cells. So that's activin concentration is so less that neither of these genes are expressed, then it would lead to the formation of the ventral mesoderm. Okay. So let me remind you about the Josh batch, which is about to start from 1st July, and we have very limited seats left. So if you wish to join this session and if you want to clear the exam, as you can see, we discuss all these concepts in class, whatever questions we are discussing. In case you are not able to answer them, and these seem to be extremely new to you, these topics, then that's not a problem, because if you join Josh batch, then you are getting, along with coaching, everything which is mentioned over here. Best thing is the test series, so that you have a constant practice of the type of questions that comes in exam, and you also have doubt solving sessions as well. And even you have access to biotechnica's online portal along with the other facilities. Okay, so you can either call the toll free number or the Bangalore number, or you can even just visit the website. Okay, just remember that it will start from 1st July. Ensure you don't miss any classes. If you want to join now, you have to enroll soon. Okay, this is a question which is based on the gastrulation process of amphibians and avians. So chick and amphibians, um, as you know, they have some homology with the structures which are responsible for the gastrulation process. Can you match the correct ones? Okay, Nandini, let's see if your answer is right. So we have the in gastrulation, we know that the pore or the opening through which the cells will start going inside, it is called as the blastopore, isn't it? So the amphibian blastopore, it is the part from which the cells will go inside, and the same thing in case of avian would be primitive groove. We know that in avians, the it is in chick. We have the discoidal cleavage, which I told you in the first question. We have the primitive streak, which arises from the posterior portion of the chick embryo. So, from the posterior portion, one small area develops. Okay, and slowly it migrates towards the anterior. There would be some cavity, some opening which would be created in this, and that is through which the cells can go inside. So, this is what is the primitive groove. It is homologous to amphibian blastopore. So C has to be 2, which means it can be either 3 or 1. Now let's see more. We know the Henson's node, it is present at the tip of the primitive streak, and this is required to organize the other tissues surrounding it. So this acts as the organizer. And in amphibians, what act as organizer? It is the dorsal lip of blastopore. So in amphibians, where we have the blastopore which is developed from here the cells will go inside. So the cells which are present near the blastopore, the dorsal lip, present towards the dorsal side, that is going to act as organizer called as Pieman's organizer, and it would specify the cells which are surrounding it. So that is why D would be equal to 4. It is the amphibian organizer. Again, it can be either 1 or 3. So let's look at something more. Now, what is the colar sickle? Colar sickle is this posterior area from where the cells will be, the primitive groove will start to originate. Okay, so it is formed at the topmost cells. Okay, so as you can see, this is the colar sickle area. This is the colar sickle. Okay, so this is the epiblast. The top cells which you can see, these are the epiblast. So this epiblast cells, the thickening which is present towards the posterior end, this is what is called colar sickle. and from there only it would initiate. So Kohler sickle is just like the local thickening of the epiblast, which is formed at the posterior area of the area pellucida. Okay, so that's why A will be 3. So A will be 3. Sorry, A will be 3. So correct answer is option 3. Okay, and primary hypoblast means it is the delaminated cells from epiblast. So what happens is, so some cells from the epiblast, it's not shown in this diagram. Whatever will be the top cells in the um, in the blastula of the avians, in the chicks, the topmost layer of cells, that will be called as epiblast. Okay, and from the epiblast, some cells will come out as delamination, and then they will join some more cells from the posterior. Thus, they will give rise to the hypoblast cells. Okay. So there is one more now. This is regarding Drosophila. Can you try to answer? Yes, Nandini, your answer is right. Very good. Correct. So this is also regarding dorsal ventral axis formation in Drosophila. We know gherkin is released and torpedo is the signal. If anyone knows, gherkin is the signal and torpedo is the receptor. Okay, so by this signal, one part of the Drosophila is specialized. So, do you remember whether the dorsal side or ventral side is specified accordingly? You have to answer. See, we know that when the uh, when the Drosophila egg is there, right? Egg surrounding it, we have some follicle cells, isn't it? And there are some nerve cells also which release the bicoid and nanos for axis specification, anterior posterior axis specification. Now nucleus of egg, initially they are present towards the posterior, but later they migrate towards the anterior, and that too towards the side which is supposed to form the future dorsal side. Remember that. Okay, and this nucleus, it releases. Okay, nature says 3. Let's see if it is correct. So this nucleus releases a signal which is called as gherkin. Okay, and the follicle cells which are present around it, they have a receptor called as torpedo, right? So that's why our option C is correct. Gherkin is active only in oocyte, that means in the oocyte nucleus, which will release the signal. And torpedo is active only in the somatic follicle cells. That means torpedo will be present in the surrounding follicle cells. So that is why C is correct. So that means it can be either 2 or 3. Now, pipe protein is made in the dorsal follicle cells. So, what does that mean? Pipe protein. First of all, you should know what this signaling is doing. Okay. So, when the gherkin signaling is there, this is the oocyte nucleus part of it, and these are the follicle cells. So, when gherkin releases the gherkin mRNA, gherkin signaling is there, they would bind to the torpedo receptor. Okay, and after binding to the torpedo receptor, they will initiate a signal which will inhibit the synthesis of pipe protein. That means gherkin signaling will inhibit pipe protein. And why is pipe protein required? You should remember this. Through a detailed cascade, which I will not discuss here, through a detailed cascade, they are going to degrade cactus and they are going to free up the dorsal. Dorsal is a transcription factor. If dorsal enters the nucleus, pi protein, if it is present, then it would degrade cactus through a signaling pathway and dorsal would be free to enter nucleus. Now, if dorsal enters nucleus, it will activate the ventral specifying cells, specifying genes. Okay, it would activate the ventral specific genes. So, don't go by the name dorsal. You might think dorsal gives dorsalization. No. The answer is dorsal gives ventralization. Okay, to activate the ventral specific genes. But where is this possible? In the ventral side. Obviously, ventral specific genes should be expressed in the future ventral side, where pipe protein will be present. Now, at the dorsal side, pipe protein is inhibited because of Gherkin signaling, right? So, there the cactus cannot be degraded and their dorsal will not be free. So, dorsal will not be able to enter the nucleus at the dorsal side, because of which ventral fates will not be there and cell will go for dorsal fate. Okay, so that's why the pipe protein is made in the dorsal follicle cells. So that is wrong, because pipe protein is not made in the dorsal follicle cells. Pipe protein will be inhibited in the dorsal follicle cells. So D is wrong. So D is wrong means this cannot be correct. So option 3 is the right answer. Yes, nature, very good. That's the right answer. Okay, option 3 is the right answer. Now let's see, Gherkin protein moves along with the oocyte nucleus and signals follicle cells to adopt ventral fate. Just now I told you Gherkin signaling will inhibit pipe synthesis and pipe will enable dorsal to enter nucleus to give ventral fate. So Gherkin signaling pipe protein is not there means it will give dorsal fate, not ventral fate. So since it is saying ventral fate, A is wrong. Yes, Alka, that is right. Option 3 is right. Okay. And why B is correct? Because maternal deficiencies of either gherkin and torpedo. If neither of this is present, if either of this is not present, then pipe protein will be synthesized. And pipe synthesized means cactus is degraded and dorsal is free to enter nucleus to give ventralization. So that would cause ventralization. B is right. Highest concentration of dorsal is in the dorsal cell nuclei. No, dorsal would be free in the ventral side, so not in the dorsal side. Okay. There is this is the explanation. You can read it later from the recording. Okay, try to answer this. This is also regarding Drosophila. This is a very easy one compared to what the level of questions that we are discussing today. So as you can see from developmental biology, if you don't have the proper basics, it is difficult to solve these conceptual questions. They are conceptual, analytical, experiment based, and there are so many different topics actually, and none of the topics are as such related with each other. As you can see, these are like separate concepts. Like in biochemistry, if you take unit 1, we have some related concepts. If you know carbohydrate, you can have a understanding of carbohydrate metabolism, but here that's not the case. You have to remember each and every little detail. So that's why you must have a proper source where you can get the proper concepts, and that is through biotechnica classes. So I will encourage you all to go through the Josh batch details, which is the upcoming session for clearing December 2024. In case you are not well prepared for June 2024, then definitely you can aim for it. You will also get double success guarantee. In case December also you're not able to clear, then you can also attempt for June 2025. Okay. Okay, option 3, everyone is saying. Let's see, Anupam Nandini, whether your answer is right. Yes, so we know that these are the mutant phenotypes for the gene. If bicoid is not there, we all know head and thorax is deleted. So D will be 3. There, you all, nothing to solve. Okay, so as I said, it's the easiest question. Even though if it is a part C question, it is one of the easiest. We know that Oscar is responsible, it's acting as a scaffold protein for binding with nanos, and nanos is in the posterior side, which gives rise to abdomen. Okay, so that's why Oscar is not there, means there will be no abdomen. So C will be 1. And torso is a terminal protein. Torso like protein will activate torso, and torso will be repressing Groucho, because of which terminal specification will be done. So A will be 4, no termini, and Vasa, it is responsible for oogenesis. So that's why if it is not present, oogenesis will not happen properly. So next question is also regarding Drosophila development, but this is regarding segment polarity genes. We know that Drosophila body has segmentation. So segmentation is done in the cellular form. Initially, the Drosophila body, it is in the syncytial form, syncytial blastula, where only nuclear division happens and not cell division. Later, the nucleuses, they will migrate towards the periphery. Okay, and later they will be covered by cell division. And then in between these cells, there would be some kind of signaling which will help specify the segment polarity. So we know that by the end of the pair rule gene expression, we would have some segments in the Drosophila body, segments and parasegments. Okay, so what is true regarding engrailed and wingless, which are all segment polarity genes? Hedgehog, cubitus interruptus, armadillo, these are all segment polarity genes. They have an extensive reciprocal type of signaling. So, one cell will release one type of molecule, and the other cell will receive that molecule. In turn, it will secrete another molecule which is going to maintain the secretion of that other molecule. So, it is a reciprocal signaling which happens. Okay, so as I will just show you since time is less. So this is the cell from which we have segments and parasegments. As you can see, this part is the segment, one segment of the Drosophila body. We know that parasegment is the posterior part of the previous segment and the anterior part of the next segment. Together it forms a parasegment. So here we have the parasegment portion starting here. So that means you can see this is the parasegment area. Engrailed, the cells which are present in the posterior end of the parasegment, we have these cells which are making engrailed and hedgehog. Okay, so these cells will be releasing engrailed and hedgehog, and they would bind to the cells just anterior to them. So that would be the posterior of the segment. So you can see around segment some posterior portion of the segment. Okay, so actually, sorry, engrailed is present in the anterior portion of the parasegment, because this is the parasegment, this is the parasegment. Okay, so this is the posterior of the segment, but in parasegment it is still here. So this is the next parasegment. So engrailed would be expressed in the anterior part of each parasegment, but posterior part of each segment. Where it is expressed, you should know. An engrail gives signal to the cells present near them, okay, and those cells will be releasing wingless. Then wingless only goes and binds to these cells. So where is wingless expressed? It is expressed in the posterior portion of the parasegment and somewhat in the middle area of the segment. These all you should know. Wingless specifies posterior of the parasegment. Engrailed specifies the anterior portion of the parasegment. Okay. So wingless will bind to frizzle receptor. It would activate the DSH. DSH inhibits ZW3, which is the GSK complex in amphibian type. And because GSK is inhibited, that is why armadillo cannot be cleaved, which is beta catenin. So beta catenin can enter the nucleus and activate the transcription of engrail and hedgehog, both. However, hedgehog will go and it's a secretory protein, goes and binds to the cells near them, where the receptor patched will be receiving them. It activates smothen protein. Smothened will activate CE or GLI, cubitus interruptus, and a part of the C gli act as transcription factor. They would enter the nucleus and transcribe wingless. Wingless will be released out and the same cycle continues. So based on this, yes, wingless is a secretory factor since it is released out of the cell. Engrailed is a secretory factor. No, as we saw, engrailed is not released out of the cell. Hedgehog is released out of the cell and it forms a long range concentration gradient. So as you can see, hedgehog, it is not giving long range, it is short range. It is only going and binding to the cells which are present near them. But yes, we do have long range in the sense the cells can go and the hedgehog can go and bind to the cells to the extreme anterior of the segment also. That is why we would say hedgehog has long range. But yes, the expression of wingless will be only in the cell which would be receiving the highest concentration of hedgehog. So in that way, this diagram is given over here. Don't go by this diagram, okay? So yes, hedgehog is a secretory factor and forms a long range concentration gradient. So it works in concentration manner, means whichever cell receives highest concentration of hedgehog will release the wingless. But engrailed is not a secretory factor. Engrailed, it should be hedgehog. So B is wrong. Engrailed regulates wingless. That's correct. Through release of hedgehog, engrailed will regulate the release of hedgehog, which forms a short range concentration gradient. So regulation of wingless is short range because it is going to activate only in the cell which is just close to them. So C is correct. Beta catenin homolog is a signaling molecule upstream of engrail. So as I told you, upstream of engrail means it is activated by armadillo. Armadillo only is beta catenin. It's a homologue of beta catenin. Okay, so it is by engrail which gets cleaved by GSK homologue. That is correct. GSK homologue is ZW3, which we saw. ZW3 was supposed to cleave armadillo, but because the DSH is inhibiting ZW3, that is why beta catenin can enter the nucleus and activate the transcription of engrailed. So D is also correct. C, cubitus interruptus is an intracellular signaling protein, signaling molecule in the engrailed expressing cell. No, cubitus interruptus is required for wingless expression. So that's why it acts as a transcription for release of wingless transcription factor, not for engrailed expressing cells. So that is why E is also wrong. Okay. Okay, then we have this mammalian one development. You can take up this as a homework later. I want to discuss one more question. So this is regarding the sex determination. Yeah, you can try this one. Sex determination of the sex determination of the human and Drosophila. So I will tell you this one. What happens over here is that the X by A ratio, which is X chromosomes by autosomes, this is how Drosophila sex determination is determined. Okay, so if the ratio is 1 or more than 1, it would form meta female, and if it is 1, then it would form female. Okay, so that can be either 2 by 2, or it can be 3 by 3, or 4 by 4, because Drosophila may have more copies of autosomes and X chromosomes. Okay, so whatever is the ratio, if it is 1, it would give rise to female. And if it is more than 1, like 1.3 or 1.5, then it would form meta female. And if it is zero point 0.5, then it will form male. Okay, so that would mean that, let's say if you have 1 and if you have 2 autosomes and 1 X chromosome, then the ratio would be zero point 0.5, which would give rise to male, right? Whereas in humans, we have the sex determination, XX will give to female and XY will be making the male. But here in the question, they have said the individuals considered in these questions are having two haploid set of autosomes and no Y chromosome, which means no Y chromosome means for humans, that means with A, you will not have female. So wherever you have A2, because they have already mentioned no Y chromosome, so that is why A cannot be 2. So wherever you have A coming to 2, it shouldn't be the correct answer. Okay, sorry, very sorry, A cannot be male. Okay, so A cannot be male, so that's why wherever you have A1, you can strike off that answer. So you can see here you have A1, so this cannot be answer, entire thing cannot be answer. Then again A1, do we have here A1, yeah, so here also we have A1, you can see A1, this also won't be the answer. So 2 and 3 is gone. Now you have to select for the Drosophila. So we know that if the zero point 0.5 is Drosophila, 0.5 ratio, it would form male. Okay, so 1B1, 1B1 should be there. Now if you check over here, we have 1B3, but here we have 1B1, so that is why 1 is the correct answer. Don't get unnecessarily confused. If you know the concept, you can easily answer. Is it clear to you now, Anupam, Yogita? You people got it, how I solved this by elimination, right? Because it is mentioned there is no Y chromosome. That means this human A, it cannot be male. Without Y chromosome, it cannot be male. So you cannot have A and 1 combination. So in these two options, you had A1 combination. Here also you had A1 combination. So strike that off immediately. And Drosophila, just now I said, if you take X by A ratio, it would be 0.5. So 0.5 means it would be male. So B has to be male. Okay, it means 1, B, 1. If you take 0.5, it would be 1. So 0.5, 1, B, 1. So 1, B, 1, it is present only in this one because here it is mentioned 1, B, 3. That is why this is also not correct. Let me explain the other ones to you. So in humans, if we have the ratio 0.5, what would that mean if you have the ratio 0.5? We know that we have this no Y chromosome. Okay, and we know anyways we have two haploid set of autosomes. Okay, we have haploid set of autosomes. So let's say 0.5, Anyways, we have two autosomes if we take X by A ratio. Okay, so if we want it to be 0.5, that means only this has to be 1. Then only we can get the answer as 0.5. That means for 0.5 in case of humans, only one X chromosome should be there. And only one X chromosome means no bar bodies because one has to be activated. So that means 0.5 humans, and anyways, this is female only. So it will be 2 without any bar body, 0, because there is no, there is only one X chromosome. Okay. But if you take 2 ratio, now X by A ratio, if it has to be 2, so anyways, we have autosomes 2. So if we want the answer to be 2, the ratio to be 2, we should take a higher numerator. That means we should take 4. Okay, X should be 4. Then only if autosomes is 2, you will get the ratio 2. So if A has to be 2 in humans, it has, if the ratio has to be 2, which means you need 4 X chromosomes. Okay, so 4 X chromosomes means it would give rise to meta female, isn't it? It would give rise, it would give rise to female only, not meta female, because anyways, Drosophila, it would be meta female if the ratio goes more than 1.5. I said, right? If it goes than 1, it would be meta female. So 2b, for Drosophila, 2 ratio should be meta female. So 2b3, right? So you can see 2b3. And in case of humans, it would be anyways, A would be 2. So anyways, A is 2 over here, you can see. But we are talking about the ratio 2. So 2A we should be looking for. Okay. So we have 2A, and anyways it is 2. So that is the ratio of the females. Okay. Now here, if it is 2 ratio for humans, we have 4 X chromosomes, which means only one will be active and the other 3 will be bar body. So in that case, if you have 2A giving rise to female, we have 4 X chromosome, then the bar body should be 3. So 2A to D. That is what we have over here. And anyways, for Drosophila, whatever you take, whether you take this, this and this, okay, or you take this, anyways it would be 0, because in Drosophila you don't have the bar body concept. Okay, so you can see for both of them, B1B1A and 1B3A, because in the fruit fly, Drosophila melanogaster achieves dosage compensation by increased transcription of the single X chromosomes in males to a level that is similar to the 2X chromosomes in females. Okay, so that's the questions I wanted to discuss. Okay, all the best everyone, and in case you're not able to clear this June exam, then don't worry, Biotechnica is there for you. Josh batch, it starts on 1st of July. Enroll soon, because very soon the batch capacity will be over and we won't be able to accommodate you, so hurry up. Thank you.